Good code music. It just fills me with sadness.
Cool.
What did I miss?
Nothing. We're just,
getting started.
not even started yet.
Okay. You know, it's Father's Day. Odd time to have a hackathon.
It's true.
No, I
Maybe
think it's perfect.
Maybe it's the
You can
best
tell the
day.
code, I am your father.
I'm gonna be in and out while I make pancakes and all that other stuff.
Aren't pancakes supposed to be made for you on a day like this? Isn't that-
That's true. Isn't that true, Sharon?
Well, I already told you I make them.
No, she will. I'm just.
But he needs time to eat them.
It's my tradition on Sunday to make pancakes, but yeah, I'll have to eat them. That will be my chore.
Well, nice of you to join us. Hello, Porg.
Hello.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah, that's going to be the limit. All right, Travis, maybe we should get started.
Okay. One second. Gopal is asking. Okay, so the call is actually full now. Is it with 10 people?
It might be.
Okay. And the YouTube link is just when you go to the event page?
Well, there's the event page, and then there's also a separate YouTube link, which I've also posted on the event page.
Okay, perfect. I'll just go there.
Now folks should be set on that.
Okay. All right. So welcome to the OpenWorm Hackathon. It's good that you can all join us today, especially on the weekend and especially on Father's Day. So that's really good that you're here. So I guess to get started, we can just go around and introduce ourselves. And since the call is limited to 10 people, we can introduce the people that didn't get a chance to get in this call at the start of the second hour. So I'll start off. My name is Travis Jacobs, and I'm the Google Summer of Code student for OpenWorm over this summer. And I'm basically working on integrating the ion channel models and upgrading the ion channel models that OpenWorm uses. So I guess that's enough about me. So on my screen, the next one to my left is Vahid. Do you want to introduce yourself, Vahid? Vahid? Okay.
Hello everyone,
Okay.
My name is Vahid, and I'm working at the modeling on channels. It's about 3 or 4 months that we have started this. Sorry if I'm it's not stable enough. Maybe I get disconnected anytime. And if it okay, Travis, can you continue on this?
Okay, I got most of that. What exactly do you want me to continue on? Oh, with the introductions? I think we got a little connection issue. So yeah, we'll move on for now. Go to Srijoy.
I'm saying I have the anytime low bandwidth problem again.
Okay.
And if you can do the interaction, I'll take.
Okay, totally. Yeah.
Okay. Hey,
Okay.
I'm Srijoy. Let's see, I'm in Vancouver, in Canada. I am the founder of Neuroelectro. I haven't I'm not actually I haven't contributed actually directly to OpenWorm yet, but I'm very interested in channel models and trying to get data about ion channels from the literature. So Steve had asked if I was interested in joining in on this call, and I was like, yeah, maybe. I also work with Rick quite a bit, so yeah.
Okay, excellent. Thanks for joining. Rick?
I'm Rick Gherkin from Arizona State, and one of my roles here is to work on model validation with data. And so what I started doing is using a Python package called SciUnit that I developed, and to automate the process of validation testing for model components and subcomponents. And today, I guess I'd like to work on refactoring some parts of the OpenWorm, what's currently being used, and then also parts of some of the channel analysis code that Porg has contributed through PyNeuroML, so that I can better incorporate that into ion channel model validation in OpenWorm.
Okay, perfect. Thanks, Rick. Porg? I think you're on mute. Okay.
Okay, fine. Okay, my name is Paul Gleason. I'm based in University College London, and I've been working on NeuroML and various initiatives around that, including open source Brain for sharing NeuroML models, and have been building some models at different levels of detail for OpenWorm using the C302 framework that I might let you talk a little bit about later.
Okay, thank you. Stephen?
Hello, I'm Stephen Larson. I'm in San Diego, California. I'm currently project coordinator of OpenWorm and co-founder with lots of other folks. I'm excited that each and every one of you are here, and if you're watching on YouTube and weren't able to make it in the channel, apologies again. But don't worry, we will be mainly interacting on chat, I think, for the bulk of this anyway, so you will be incorporated and included. But I know that Andrei Polyanov, who's also a co-founder, is watching and would like to be in the room, and potentially also, I think, Gopal Sarma. So both you guys, and if there's others, just let us know on the chat there. But they're also with us here today, so good to see you all.
Okay, perfect. Milad? Milad? Okay. Must be some kind of lag or something. Okay, that's fine. We'll come back to Milad. Joe, you're next on my screen here. You want to go?
Sure. I'm Joe Bowen. I'm a software engineer. I've been working with Python for a while now. I've been doing a bit of Django stuff. I'm kind of new to Django, kind of winging it, but been working on some of our Django for the channel worm and for movement validation as well. Kind of putting that together and getting some APIs going and that we can interact with and store data basically.
Joe, I haven't interacted with you much. Maybe I can comment on one of the issues. Srijoy and I have both done some Django stuff for different websites, and I wanted to. It's not something I want to be involved in a lot otherwise, but for the purpose of the hackathon, I would like to just volunteer if you have any thoughts or stuff that you're wondering about. I have a fair amount of experience with Django.
Okay. Yeah, definitely. I'll pick your brain if I have anything right now. I'm not sure what kind of questions we have, but I'll let you know.
Okay.
Okay, perfect. Okay, next on my screen is Chiwai.
Hi everyone, I'm TYB. Currently unemployed, but I have background in computer science. My dissertation was on parallel computing, performance tools, and scaling parallel applications. So I've not yet contributed to any part of OpenWorm, but I'm interested to figure out like what parts fit together, how it works together, and personal interest is in trying to measure performance and also to scale any parallel applications. I used to try to scale stuff to hundreds of thousands of processor cores, so that's going to be fun. But first, I guess just here to try to figure out what the pieces are, and also the kind of workflow and work structure that's very different from academia that I was used to. So yeah, that's pretty much on here.
Okay, thanks, Chiwai. And last but not least is Abdullah. Actually, we'll come back to Milad after Abdullah as well. So Abdullah, you want to go?
Sure. I'm Abdullah Rashid, and I'm in Lahore, Pakistan right now. And I've contributed briefly to the C302 project, and I'm looking forward to contributing again today.
Okay, excellent. And if you can hear me, Milad, do you want to give a quick introduction? Okay, maybe you can't hear me. There might be some kind of connection issue. Okay, that's fine. And there's a few other people as well that are watching, and if we get a chance, we'll give everyone an introduction as well. And also in the Gitter chat, you can introduce yourselves if you'd like. So basically, I'll just run through the logistics now of how this Hangout's going to work. And then we'll give a short presentation, and then we'll actually get started with the hacking. So first, I guess, with regard to logistics, we're basically going to, after the presentation that Stephen and I give, which is sort of a summary of what's been going on lately in OpenWorm and the areas where you can contribute. After that presentation, the idea is that you guys will kind of get a sense of what's going on and find a place that seems appropriate and of interest to you where you can contribute. So that on the agenda, that's called the Birds of a Feather breakout session. So everybody who's interested in similar aspects of the project is going to get together and work on that piece in their respective chat room. Also, the agenda should be available on the event page. But if you'd like, if you can't find it or whatever, you can ask me on Gitter chat, and I think Steven's controlling the Gitter chat as well now, so you should be able to find it there. Okay, then beyond that, we have a Trello board. I'm on the agenda right now, actually, if you want to run down through it with me. And actually, what's best for me to do is to share my screen. Just give me one moment. Okay. So right, I just spoke about the breakout sessions. So we've also got the event page. That's where this agenda is posted, and there's links to the YouTube viewing of this Hangout and also the Hangout itself. Then we've got the Trello board, which summarizes all the milestones across repos and across sort of sub-projects of OpenWorm. So as Stephen and I talk about what's happening lately in OpenWorm, we're going to be referring to things here. And this is also somewhere that you can comment, so you can have a look at each of these milestones, look at the title, say, I'll start at the top. So we've got, this is the repo that is involved with ChannelWorm, and this is the name of the milestone. So augment web app to contain model completion dashboard with connections to PyOpenWorm. So that sort of gives a short description. Then if you want more detail, you can go to the GitHub page, and that gives you sort of a rundown of each of the criteria for completion for the milestone, and then there's also the waffle board, which you can go to, and it filters by this milestone, so it shows you all the issues that are involved in this milestone. So we'll talk about this a bit more as we go on as well, but just to make you aware that this is available in the agenda. Then there's waffle board, which I just mentioned. Okay, so this is just loading up here. But this kind of cross-cuts several repos. Okay, here we go. Yeah, so the idea here is that we can look at issues across repos and organized by, well, however we choose. So in this case, they're organized by different labels. So issues with the ready label or the in progress label are in the respective columns, and then you can filter by different things. So you can filter by milestone. So those milestones that I just mentioned, you can pick one of those. So let's say modeling ion channels with no patch clamp data available. There's only one issue in this milestone. But you can, yeah, so if you pick a milestone that looks interesting to you, then you can come to the waffle board, filter by that milestone, see which issues are ready to be done, and then you can take one on. So this is kind of the interface for interacting with issues across all the repos that we're going to be using today. So that is the waffle board. And then we've finally got the roadmap page. So I'm actually just going to bring this up and just give you a glance. So this is, without going into any detail right now, because we're going to give the presentation shortly, this is basically kind of a global overview of how this project works. So you can see it seems probably a bit complicated. There's a lot of nodes and a lot of arrows going everywhere and different colors and everything, but we're going to do our best to explain that in our presentation, which we will start right now. Okay, I'll stop sharing my screen, and Stephen, if you want to start off the presentation by sharing the slides there.
Yep.
Okay, perfect.
Okay, guys, thanks for joining. I'm going to try to not be too long-winded here, but I just want to reemphasize one thing that Travis said, which is this birds of a feather thing. So in the agenda should be world editable access. At the very bottom, there's something that says birds of a feather. So the idea is that at any point here through this presentation, if you're interested to know more about any topic that I bring up, you should put that topic down on that list, and you should put your name next to it. If you see a topic that somebody else has put down that you are interested in, you should also put your name next to it. So, and this should be going on basically continuously while you're listening to this. So this is something active for you to do. By the end of talking, there should be at least one topic that you're interested in learning more about. Because what we're going to do after this is done is we're going to do breakouts. So this is the purpose of this as a practical hackathon is that by the end everyone's got something they're really happy to hack on during the process. And this is just to kind of get some clarity as to the bigger picture. So please go in, edit the birds of a feather, start putting ideas down for yourself. You can have more than one as well. The idea here is that we have a list that we can sort of prioritize. And then Travis and myself and others who are knowledgeable will split out and try to explain more about a given topic. Okay? All right. So okay. So we put together some slides here to try and get some clarity. So Travis just showed you all the issues, but you kind of need a bit of a big picture as to what we're doing in the project as a whole. So so the goal of the entire project, and this is sort of a sub-project of it, is a full-scale simulation of C. elegans. Oh, I should also say, by the way, feel free to use the link that is in the agenda to pull up this presentation for yourself. It should be public. And often in the bottom corners here, you're going to see underlined words or explicit links, and you should feel free to click out to that. So the entire presentation is pretty much annotated this way. So at any point, like this year goes out to the paper that we got out last year, for example. you can read more details. If you haven't come across the paper yet, should be accessible on this, and so on and so forth. So that should be another thing that you can do other than just listening to me passively. You can feel free to pull up other things up across the web as I talk about them. Okay, so full-scale simulation of a C. elegans, right? So we do that in three major ways. One is through the carrying out of open science, meaning that we're doing science in a way where we're putting out our methods and our content up online as we do it, as opposed to only after the fact through a publication, although we do that too. Through engineering, that's where hackers and programmers from around the web and just folks who like to build stuff come together. Mainly this is software engineering. And then also by building community. So by talking to folks in the context of journal clubs, or going out and physically meeting people at meetings, or having hackathons like this. All right. So one of the ways that I've been explaining the project recently is like, why are we doing this? One of the lenses into this is through neuroscience, and one of the ways in which neuroscience can be influenced is through AI. So these are two subjects that are sort of dear to the hearts of many in the project. And so back in the 80s, there was a sort of a bit of a revolution in AI, where they started to move away from programs that just sort of thought in a box and robots, and started building robots with the idea that the brain is a control system for the body, and you can learn about how control systems work by starting to build physical robots and trying to make a control system that does something intelligent. So this guy here, Rodney Brooks at MIT, sort of launched this whole kind of idea, and he's ended up actually building the company that built the Roomba, for example. So it was kind of a way in which, through not having any clue as to how to go about, reverse engineering a brain or a mind, it was a new approach to doing this that they called embodiment. So the basic idea of that is that you have a body like a robot, and it's the interface between a world where there's things that are happening and a system that has sensory input and control and motor output. And so this is very much analogous to what our brains have to do. Our brains are essentially a control system. They're moving around like the robot that is our body, and it has to interface with the world, and so there has to be this interaction between these things. So, so one of the original ideas for motivating OpenWorm was, could we do this with a very simple nervous system, but actually reproduce a nervous system? And if we're going to do that, we're going to need to have a very simple body. So this is one academic effort to build virtual crickets in a computer, and so we're not the first to think about doing this, but so we've sort of chosen to do this with with the C. elegans, and I'm not going to show the video here, but many of you, since you're already part of the project, sort of understand why C. elegans is interesting. It actually has interesting behaviors. It's the first organism to be sequenced. To have its genome sequence, its cellular anatomy is very amenable to this. It only has 302 neurons and 95 muscle cells. We know its cell divisions, and we know its connectivity graph of its brain. So what we've been doing in the project as a whole is basically to put the worm and the worm's brain and its into this same context where it's operating in a simulated world. We have a simulated body, and we have a simulation of neurons and muscle cells. And that's really the sub-project that we're taking up here today in this hackathon, is the process of making our cell model of both neurons and muscles much more detailed, especially as it relates to ion channels. And then, also the purpose of this, which I guess we don't talk about as much, but it's also pretty important, is that if we have that simulation, we should be able to start extracting some mathematical principles from that nervous system running as a way to try and actually, get a deeper sense of how neurons in general work and how a circuit of neurons function in a way that we can potentially get to engineering a circuit to do the kind of control that we would want. That's kind of the grander goal on the neuro side. Of course, there's all sorts of interesting things even on the genetic side, things on the development side. So there's lots of other components of this project, but this is one that sort of was some of the original focus and the one that I think concerns this subgroup the most. Okay, so our long-term goal is a full-scale simulation of C. elegans. Our medium-term goal is accurately predicting a database of actual worm behaviors by using a data-driven, cell-by-cell 3D neuromechanical model. And so this is kind of just connecting you guys to other parts of the project that are going on. So there's another whole sub-team that is working on this problem, which is how do you take videos of real worms that have been recorded and segment them using computer vision, and then extract statistics from them. So in order for us to not build a model that's just kind of an analogy to a worm, but one that actually matches some reality, so we have to have actual data. So there's been a whole sub-team that's been sort of sharing open source code around doing computer vision in this way and extracting statistics, and they're doing a really great job. Now, on the other side of that is the work that's been done by Andrei Polyanov and Sergei Khairulin, and that's to build a mechanical, the virtual mechanical model. So we actually have this. This is what's called cybernetic. It's under the Smooth Particle Hydrodynamics repository, and this is our sort of virtual particle simulation cockpit, if you will, for the nervous system. So if we had our nervous system model with all the neurons firing in a way that we were fairly certain was data-driven, we can start to plug into this and actually have little virtual muscles that are pulling. Right now what you're seeing is a sort of sinusoidal input that's been put in here, but we have the capacity to do that. And this also gives us something that we can do the same process of segmenting the shape of the worm and comparing it to what the real worm does, as you saw in the other video. Okay. So then this just shows that, right, so we would, so our process that we are building, our pipeline, is that we use computer vision on movies, we extract features, we also extract these same statistical features from our model, we compare them, and then we get some kind of a score that tells us how close our model is actually fitting to real behavior, and then we use that as a way to improve the model and get it to fit even better. And this is the sub-team that's headed up by Michael Curie and Jim Hokinson, but also involving Andre Brown, who's been very generous in providing us this data, as well as William Shaker. Okay, but now coming down to getting closer to what we're talking about here, it's worth pointing out, and we've talked about this at length on a whole journal club. So if you want more details, you can check out our YouTube page, and there's an hour and a half talking about this subject. But as it turns out, we still don't know how the worm's nervous system creates the crawling behavior, even the simple sinusoidal crawling behavior. It is not known in science. And this paper here, if you click on this link, you can get out to this paper. Even as recently as 2014, there's reviews that have multiple theories for how it might work. But none of them have been definitive. So this one here, for example, says maybe there's an oscillator in the head and the neck, and then through the neurons that go down the body of the worm, there's an oscillation that's then propagated, but there's no active oscillation happening down the body. Another one says that there's actually oscillation that's happening, active oscillation that's occurring through circuits all throughout the body in this way here. And then a third one says that there's not any oscillator. There's just interactions between the sensation of the worm that it gets from the outside world and circuits that are inside. So it's a loop of sensory and motor that's happening sort of at each part. And so nobody knows. Nobody knows the answer to this. There are a few researchers, like our collaborator Netta Cohen at Leeds University, thinks it's sort of a combination of some of these, strongly thinking that proprioceptive input is a big part of this, but it's basically a mystery, and this is where it gets cool for us here at OpenWorm, because in the building of this model and the way that we've been doing, we can potentially try it out all three different ways and see, is it possible that it works one way or the other? What are the constraints? So that's largely where we're headed with this, and then of course to plug it into this and see if physically it makes sense if the pulling is happening on the muscles in the right period. So in the time that OpenWorm has been operating, we've actually got a lot that we've built up and a lot still in motion. but fundamentally we're driven by these sorts of scientific questions, and if we can say something about them, then we want to get that out to the world and publish that. And I think that's one of the things that's really exciting that we can all kind of do together. So, so that hopefully shapes a little bit of the scientific context. As several of you know as well, in the early years of the project, we put a lot of effort into building this connectome, which is both using the atlas coordinates of the C. elegans, as well as has the connectivity graph in it, as well as reusing technology called NeuroMel, which is a really great way for representing neurons and sharing computational neuroscience models. And you can see all that on open source Brain, and as well it's backed by repo that's in the OpenWorm project. So this has been the effort of several folks, and but as soon as we built it, we knew that it was incomplete, and part of the project has been to flesh it out more. One of the things that is incomplete about it is the kind of ion channels that are associated with these cells. So, so there's challenges on the computational neuroscience side. There's a lot of parameters that have to be set with regards to the ion channels, their density, that kind of thing. We know from the field of computational neuroscience and a researcher in particular named Yves Marder, very famous computational neuroscientist, that having a lot of parameters means that the model could be right in many different ways. So that's always a challenge. We can test the dynamics, though, of individual components of a complex multi-scale model in code, and that's a very helpful thing. And this is where frameworks like SciUnit, which we'll talk a little bit about, are very important, because you could very easily build a model that just has a million different possible ways that it could work, and you never know which one is even close to right. So, so we want to make sure things like, well, when we run the simulation, we want to make sure that individual parameters are within biophysical ranges, so that we, we don't have part of our model be completely out of the realm of physical reality. We want to make sure the combinations of values are going to result in dynamics that are close matches with experimentally reported data. So a big part of what's driving what we're doing is matching data that has come out in the literature. And and so like I said, I'll talk a little bit about SciUnit as a framework that Rick Gherkin here invented. So it's really great to actually have him here. And we've started to use it in the project as a way to basically constrain our models and give us a solid foundation that we can both point to, but also let the community kind of kick the tires and decide for themselves if, in fact, the models that we're building are matching reality or not. And the beauty is that it's all on GitHub, of course, so folks can check that out. Okay, I also wanted to talk a little bit about the third sub-project that's currently being managed as a sub-community, which is called Geppetto, also relevant to us. So Geppetto is a web application, helps you control biosimulation through a web browser. You can check it out at geppetto.org. You can see a live version of it running at live.geppetto.org. What's really cool about Geppetto is that anything that we build in NeuroML, any of the channel models that we build, any of that stuff, we can directly load it up into this live.geppetto and we can see it running. It's actually being expanded to do a lot of other cool stuff. This is also a component of open source Brain. So what it basically lets you do is, in addition to just looking at the structure of your model, you can actually see what it's like when you run the model, and you can see the dynamics of it, both in 2D, as you can see here, and as well any three-dimensional structure that's associated with it. And a lot of energy has been put around making this happen. This is an example of drilling down and being able to see, the underlying equations of a Hodgkin-Huxley model. And in this case, you can see a connectivity for a larger network. And so for us, this is going to be important when we load up our C. elegans network in this connectivity graph, which you can already do the 3D part. Probably we can do the 2D part very easily. But it's a way basically where everybody can explore the same stuff and not have to build everything completely from scratch. So for us, for those of you who are here who are more on the programming side, we're probably more comfortable building things from scratch. So that's great. But the way the place this really comes in is in OpenWorm's mission to do open science. And what we find is that it's open to a degree if the source code is open, but it's a whole other thing if folks can easily get at stuff over the web. So about a year ago, we did a Kickstarter to make it so that we could put the contents, so we could use Geppetto as a backend and create something called WormSim, which puts a model, which puts the worm model to date up on the web, so folks didn't have to build it. And so actually, this is the other big thing that's happening in the project, is that this is going to be released in the next few weeks. We're kind of coming down to the end of this, and it basically puts this browser interface on OpenWorm. It lets folks in the community drill down into cell activity, gives you a web console, and so for the first time, folks are going to be able to see a more dynamic view of the worm that's operating in here. And I mention this because this is a vehicle for all the effort that all of us are doing to get out into the world. And we've actually been coming up with some milestones which now have the awesome ability to basically say we want to design a model, we want to design a component of the model of the C. elegans, and we want to push it all the way out to WormSim so that anybody in the world is able to see it. This is something that we're connecting with classrooms around the world, as well as students, individual students, I should say, so both individual students and students in classrooms, to be able to learn about the C. elegans. It's going to be really exciting when this launches. I guess I just want to point out here that the product of what we are doing as a community, it's great when it lives on GitHub, but it's even more exciting when it can make it all the way out to people around the world, no matter where they are, just through their browser, which is a very low barrier to entry. So just as a way to kind of, that's kind of the full sort of circle and the arc of this, all the way from an idea of something cool to do, answer some scientific questions, but also make that available to folks. So we really have something that's people-powered computational biology, both from the designer side, the scientist side, the programmer side, and the education side. Okay, many of you here are calling in from around the world. This is just a small sample of folks that have contributed to the project, but you can see where some of the arrows are here from around the world. It really is a pretty international project. We've also had folks from South America as well. Also, I think in this particular picture, Western Europe is a bit shortchanged. We've had folks from there as well. But you get a sense of the scope of the project. There's lots of folks operating from many different areas. The DNA of the project is both from producing open source software and inspired by open science. These are two great references, by the way, if you're curious to check out and learn more. And these are contributors over time and different organizations that are involved. Okay, so now getting a little bit further down here. So you've all been working on GitHub. Recently, I pulled this up for a presentation that I did for Oregon Street over there, and realized that across the whole project, we have closed 931 issues on GitHub since we moved to GitHub, probably at like the end of 2012 or beginning of 2013, which is awesome. And at the time that I did this presentation, it was a week or so ago, there's 337 issues that are open. So, and there's 45 repositories. So on the one hand, it's great that we've done all this work. On the other hand, folks like you who are eager to do stuff are probably dumbstruck with the number of possible things that you could do in the project. And so this hackathon and this presentation is an attempt to kind of clarify what at least one part of the project is up to, so that you can have some accessibility into what that is. So as Travis was showing you, we rely on some tools to help us visualize all of these tasks, and Waffle is one of them. We are making an extra effort to come back and use this tool, which we, sometimes, so Geppetto uses it really well, the Geppetto subproject. I haven't used it as well personally, but we're trying to get back to that because at the level of creativity and collaboration that we want here, we really need to have something that can scale up on this. So basically, on one page, this is about what, like 15, 20 different issues that folks can work on, and it's in the context of a Kanban board, which basically is just tasks that are in columns, where you have a backlog of things that could be worked on, things that are ready to be worked on or folks have committed to, work that's in progress, sometimes things that are needing review, and then a list of things that have gotten done recently, which is also good just to see our progress. Also, in case you aren't connected to us, we're pretty active on Twitter. The project actually started with a tweet. If you didn't know that, it was a tweet back in 2010 between myself, who was managing this Twitter account, and Giovanni, one of our co-founders, who was basically like, "Hey, let's do the C. elegans brain," and then I was like, "Yeah, let's do that," and then a lot of other folks came on board after that. So, so we're big on Twitter, and then our YouTube channel. I would also just remind you. So we're now, all this that we're talking about here is out on YouTube. There's a lot of, it's a good way to keep in touch with the project, and also there's a whole list of journal clubs that go into a lot more of the specific details about the science that we don't have time to cover right now. And over time, so folks like you have been volunteering for the project, and in the first few years of the project, it really was very flat, but this has been increasing. And so several of you who joined us in the January and February journal clubs, or muscle neuron modeling groups, we're doing good stuff. I think the challenge of this whole arc where folks are going up is an organizational one. And so today this hackathon is also an experiment for us to figure out how we can work collaboratively, where not every single person has to ask me what possible thing they can do, because as it turns out, that doesn't scale up. So thankfully we have Travis here, who's coordinating the project, this project for the summer. But also we hope to make it so that you guys have enough of the vision of what we're trying to do, so that you can actually contribute your ideas of what a good next milestone should be. That's really why I'm spending the time to explain all this to everybody here, is because this is not just sort of my scientific idea, or like the scientific idea of those folks who've been in the project for a while. This is something that everybody should kind of have the general sense of what we're trying to do, and everybody's free to kind of like pick up a part of the project and run with it. That's the whole idea. So that's the only way I think to sustain this growth in folks that are coming in and checking out the project, is that if all of us have something to say about what we're doing. So okay, so details. All right, so this picture, and again, I hope if there's anything in there that you want to know more about, this is an opportunity for you to take a minute, write that down on birds of a feather, and for us to have a session to discuss it later. So hopefully that list is getting populated. I haven't clicked over to look at it. Okay, so this roadmap, so this is now for a piece of the project. This is going to go from these high-level abstract ideas down into things that actually relate to specific repositories that are in GitHub right now. And so let me sort of start with this flow here from the left. So the project is composed, this sub-project is composed of the major repos of ChannelWorm, PyOpenWorm, the muscle model repo, an effort that's gone into that, and C302, with other contributions from the Neuron optimization package, as well as a decoration of tests that are built on top of the SciUnit framework. Okay, that's the basic picture that we have here. And there's sort of a flow that is represented in this diagram that kind of starts with the ChannelWorm pipeline. And so the ChannelWorm project is being spearheaded right now by Vahid and Milad, as well as Travis, sort of working in collaboration. And it's a very sensible place to start, in that ion channels are the foundation of the electrical dynamics of both muscles and neurons. So the idea has been to take scientific papers about ion channels that either have patch clamp data or don't have patch clamp data, and to extract the figures from those papers where there's key data about the dynamics of an ion channel, and then digitize those, and then create models using the NeuromL2 language for defining ion channels, and then fit them. So the fitting part is something called optimization. So this package called Neuron has been built to do this. I think Vahid already has this loop working, so that we are able to do this, which is pretty cool. Something called CW Fitter is responsible for that. So if you're curious or interested in the process of fitting ion channel models, I know that's something that's is work that we need doing. Anyway, so when the model doesn't fit, it goes back, it gets optimized again until it does fit, and then we start to build up this library of ion channel models. We know, if you're not a biologist, ion channels in worms and ion channels that are in your brain, they're essentially the same thing. So there's several differences, but essentially it's one of the reasons why neurons in the worm and neurons in your brain kind of work the same way, because they're based on principles of dynamics of ion channels. Alongside this are tests. So I think the big idea that SciUnit puts forward, and again, Rick is the father of this on Father's Day, and he'll tell you more about it. But the basic idea is if you have a model and it's supposed to fit some data, there should be some rigorous way to ensure that that's true. So the whole idea of SciUnit is, in our context, is that we can build a test that says if you're an ion channel, you're gonna have a time series that looks sort of like this, or you're going to have a current voltage relationship plot that looks sort of like this. And to be able to put this out on GitHub, to have it be run automatically by Travis CI, for example, and generate a plot, is a really really good way to make this transparent. And since this is a project that's very complex, very hard to know when something is accurate or not, having a lot of tests that do that is essentially reproducing what we do in software engineering, which is that the only way to kind of manage the complexity of a giant software application is you have a lot of unit tests. So this is the same idea, but for models. Then those models are distributed out to cell models, and so we have two major repos that do cell models. One's the muscle model repo, where we're modeling one muscle model in high detail with NeuromL2 and populating it with ion channels and making sure that it works well as C3O2, which is focused on the network, on the connectivity patterns, on the neurotransmitters, and that kind of thing, but which also increasingly needs to be gaining ion channel models and be populated with that as well. And you can see that there are also blocks here for tests here as well. So does the muscle model do what we expect the muscle model does once it's populated with ion channels? And then various tests on the C3O2 network as well. And I will say, for folks kind of getting into this stuff, we have a lot of milestones right now that are focused on getting these tests up and running. We're kind of starting to go towards at least either a test-driven development or like test parallel development, where these tests are getting made. These tests are getting made maybe even before we make a progress on the model itself, so that we know that that content is there. And these are things that are kind of relatively easy for folks to get into. We now have templates for SciUnit tests that you can use to kind of get into this. So testing, I would say, might be a topic that could be a really good breakout session for folks. And then you can see here, it could be testing on the muscle model, it could be testing on C3O2, it could be adding more tests on the channel worm side. It's kind of a nice way to kind of get into some of this stuff. And Gopal Sarma, who's joining us, has kind of been excitedly taking on the mantle of test master, and we're really glad that he's done that. So in terms of some of the practical stuff as well, but a team of both Rick and Gopal, as well as Travis, can kind of help you getting started on how to build these tests and what tests to build and how that whole framework works. So that could be a really good breakout topic. The core here of this is something called PyOpenWorm, which is a database. It's a simple API for facts about C. elegans. We're very proud that we just released a new version of this. Actually, yesterday we announced it. We did it earlier this week, but it's actually out on pip, so you can do pip install PyOpenWorm, and you can get a hold of it. And the idea of this is that we have a lot of data in the project that's been sort of scattered across a lot of spreadsheets, and we have long wanted to kind of reconcile that all in one place. So PyOpenWorm is that one place, and so now instead of having our models be driven by spreadsheets, some of the work that we are doing is to switch over the input for these things to API calls from PyOpenWorm. And it also has its own tests, and actually the tests for PyOpenWorm, I think at this point, are probably the most numerous and probably the highest quality of tests that we have anywhere else in the project, although we haven't yet incorporated SciUnit into this part. These are sort of more traditional unit tests. I think SciUnit's more for these other more model-based stuff, but a lot of if you want to get a sense of what good tests look like, you can see that over at PyOpenWorm. And then the last component of this, which is kind of hanging out over on the side here, but we will talk about, is there's been folks that have been excited to automate this pipeline through a web interface. And so Milad and Vahid have recently put out a web app on this, and in collaboration with Joe, have actually put it up online, and so such that it's live. I think it's a great first release. I know that there are still features that need to be implemented and things that need to be added to it, but again, kind of from an open science context, having interfaces that folks can see at least gives you a clearer sense of what's going on under the hood, and eventually creates a way that multiple people can interact with it. Okay, so that's the basic idea of the pipeline. Coming in from papers, building ion channels. I should also say the papers inform how we build these other pieces, C302 and muscle models, so it's not the only place the papers go in, but for the purposes of this, you kind of get a sense. Data is either flowing in or out of OpenWorm, and then there's tests which decorate this all throughout. I should also say that the optimization right now is focused on channels, but the optimization can also happen at the level of muscle model and can also happen at the level of C302 to some extent for individual neurons that are in there. Okay, now some resources to help you understand. So several of you probably have come across Hodgkin-Huxley dynamics. I talked about it in the muscle neuron presentations originally, but I just want to point out now, if you are on the slides, that this has been compiled into a tutorial. It was originally kicked off by Joe Bowen, really awesome contribution. It's since been expanded by Travis to include even more than just a simple Hodgkin-Huxley equations example or a simple LEMS model, but now it also includes how to run some of this using PyNeuroML and how to even get down to the level of IV plots. And it also, Travis has marked it up with lots of references on the basic math. And so a lot of the topics that I covered in my first couple of sessions there, where I was talking about the math of Hodgkin-Huxley, I'm not going to go over that again. You can check out this Hodgkin-Huxley tutorial. And if you're really just coming in and you're not as familiar with that stuff, I mean, you could spend some of your time hacking on just like playing with this code and getting this to work. So this could be one thing. If you want to learn more about that stuff, it's pretty accessible now. It's also backed on GitHub, so if there's anything in here that needs improving, you can help improve it. But this is actually at a pretty high level of quality right now, as it is. So a good way to kind of get a sense if you don't understand the science. When it comes to kind of the cellular stuff, I will just kind of briefly walk through the connection between the models and the graphs. So this is the image on the muscle model page, but basically, in order to get to the place where we have muscles that are that have electrical dynamics that reproduce what biology is doing, those ion channel models that we're fitting in there actually give rise to dynamics. And we see those dynamics when we do what's called either a voltage clamp or a current clamp experiment. So voltage clamp and current clamp are just different ways that we can stick an electrode into a muscle cell. Well, we stick the electrode in a muscle cell, and we do that either way. But then on the back end with an amplifier and a stimulator, we can either make it so that we're looking at the change in voltage over time across the membrane, or the change in current over time across the membrane. And again, this is what the Hodgkin-Huxley dynamics, this is what the Hodgkin-Huxley equations basically reproduce. And so getting these traces to be fitting what real papers say they are is part of the project of what we're doing at this level of scale. So, and then, one thing that you can do, so if this is your muscle, if this little pink thing is your muscle, one thing that you can do is you can use light-sensitive channels to activate those ion channels, and you can record from them. These are all the muscle cells in C. elegans, by the way. These are the 95 body wall muscle cells, so they all look like these little, these are sort of idealized as these little ovals in this case. But so once those muscles are actually stimulated, once you can activate those ion channels, this movie here just shows like what that actually looks like. So folks have actually, used a microscope and had a look at the way that the tissue actually bends, so it just kind of deflects like this. But the point here is that the reason that these muscles are pulling is because of an electrical change that's happening across the membrane. Okay, so when we're reproducing this electrical activity, this is basically the signal that's going to cause the muscles to actually pull. So this is a paper that starts to encode that. And we have one issue right now, which is to make our muscle model kind of reproduce this. But this is a trace over time where they use light to stimulate these ion channels. The ion channels opened. This is the change in the electrical potential of the muscle cell over time, and then they turned off the light, and then it goes back. So reproducing this sort of time course is something that our muscle model should be able to do. We don't know right now if it can because we haven't written the test to do it. So that's one of the things to potentially work on. This is just showing that muscles and neurons are related to each other. You can check this out in the browser, browser.openworm.org. So going from just muscles to neurons, the key thing, of course, is that to point out is that neurons synapse onto muscles, and they also have ion channels, right? So the neurons have ion channels, the muscles have ion channels. You can also stimulate the neurons using light-sensitive channels, and you can then start to do things like study the synapse between the neuron and the muscle itself. Okay, so this paper also kind of shows what happens when you do that, when you stimulate the neurons and you get the muscles showing activity like that. So again, these sorts of traces are things that can go into SciUnit tests that can be run against the models that we have. It can actually even do some optimization as well to see if we're actually fitting with what real data tell us. So this whole process of digitizing figures, for example, it applies just as well on the side of channels as it does on the side of neurons. Okay, so that's just kind of a bit of a tour of some of the, when we say like extracting figures and digitizing them, like we're talking about these sorts of traces over time, or we're talking about this kind of a trace over time, and comparing it to what a model that's built on Hodgkin-Huxley equations are actually doing. All right, now this is also an idea that folks may or may not be interested in. I don't know, but it's this is a pure sort of prototype idea that came out of some of the discussions of how to make this even more transparent. So one thing is to see the worm moving, but it's another to kind of get a visualization of what data we actually have. So this is an idea for a modeling dashboard that is an interface that summarizes in a single image, basically, what's in PyOpenWorm, and therefore what models we actually have. So it's sort of a grid-based view where I showed you that there are the muscle cells, that are kind of lined up in rows across the body of the worm, and then there's a neural network that so this is about 302 neurons in a grid. The idea is to use color and be able to have it so that you can like roll over these different cells, and so that you can immediately see for any neuron that's there how much we know about it, or any muscle cell how much we know about it, specifically related to how many ion channels that it has, and whether we know things about those ion channels in our database. So the idea would be you'd like roll over one of these cells here, maybe that's green, and then in another panel you'd bring up that the name of that neuron, say, or let's a muscle cell, in this case a muscle cell. So let's say we're looking at MDL08. There's another grid that shows you, again, color coding on the ion channels that make up that the dynamics of that muscle cell. You can also roll over those and see the names of those, and then immediately you see a picture that's basically the result of a SciUnit test that shows you the data for that given ion channel, like an IV curve, and it shows you the result of the latest simulation running that model. You can see them overlaid on top of each other. So just really quickly visually, you can get a sense of like, is this particular ion channel model actually fitting the data that we have or not? And you can do that across 3, 4, 5 different traces as well to see maybe it's fitting better on one trace, but it's not fitting as well on the other one. And then if you want to drill down even further and you click on that ion channel, you should actually be able to get down to the level of that ion channel, and you should be able to see the same kind of graphs, but at the level of a single ion channel, which will be kind of different data from different papers and that kind of thing. And then, of course, there should be references for every bit of data that we have. There should be a list of references that show us, like, for that figure, like, how do we know where that data comes from? We can have that at this level and this level as well as this level. What I like about this is kind of in one picture defining a lot of requirements for what we need to be doing. So we need to be gathering these figures, we need to be digitizing them, we have to be assembling the ion channel models, we have to be able to run the ion channel models, we have to be able to run the tests that run those. And so if folks want to work on this, this could just be to have this visualization that pulls even now out of PyOpenWorm, where we don't have a lot of this yet quite encoded in PyOpenWorm. It's still a really good kind of rallying point, so we can all see over time like how much closer are we getting to really filling out the neurons and the muscles and really like making this whole pipeline work. So basically, this whole pipeline is finished when like all the cells here are green and all of these are green and everything in here is green and we have data for absolutely every single thing, right? That's when it's fully finished. And we know that's probably not going to happen immediately, but it can happen over time.
Stephen?
So, yes.
Hi, could I just make a suggestion or a point? I think
Yes.
in the short to medium term, I think there's still going to be a long, long way before we have individual models, or we want to aim for individual models of neurons or muscle cells. Certainly there could be different details for different types of neuron, but I think in the intermediate stage, I mean, okay, yes, something like this would be very nice to assemble all the different details, but I think in the intermediate stage it's much more useful to plan for a kind of canonical muscle model and a canonical or a generic neuron model. So, I mean, certainly, as I say, it may be the case that you'll have individual experimental data for individual neurons, but I mean, if it can be built in from the start that there will be different levels of details for different types of models, and in some initial models, there will probably be the same muscle cell model for every muscle and the same neuron model for every neuron, maybe with just differences for size and so on.
Yes. That's great, yes. So part of the idea of this presentation is to be provocative in certain cases and sort of stimulate thought. So this is one topic that we can discuss. This does not need to be the only thing that gets done. And I think the approach of having multiple different levels of detail is great. We already have that to some extent in C302. So I completely agree. Let's make that a discussion point as we go on here. I realize that this presentation is a bit lengthy, so I want to get folks to hacking. I have just a few more things to get through with this, but this is even if we don't build this thing out, this should at least be in folks' minds for potential target of what complete might look like at some point. It might be really long down the road, but anyway, I just wanted to. I think this gives some color for what the enterprise of what we're kind of trying to do here kind of looks like. Is to have a complete picture of ion channels inside different kinds of cells that exist. So that's kind of big picture kind of idea. Okay, let me get through this real quick. So so there's model optimization that helps fill in some of the gaps. That's this blue picture that's here. There's some papers that talk about this. This gets pretty techy, but if you're interested in this stuff in optimization, there's many places in which this fits into the pipeline as well. So that could be a good breakout topic. I don't have time to really talk about too many details, but basically we can use machine learning to fit the parameters that we don't know and keep them within biophysical ranges. So this is the SciUnit piece here. I think I've been talking about it throughout, but it's basically taking this idea of unit testing from software engineering, where we can run a unit test, fix some bugs every time we check code in the repository and check that code out again, make changes to that. We're continually running the tests. So every time the models are updating and changing, there's a test which is telling us if we're still within the ranges that we want and that kind of thing. And I think it's a really smart way to deal with this challenge that there's like a million different ways that you can build a model. This is a loop that we can talk a little bit more about as well, specifically in the channel model stuff. So this is just a little bit more specific than that giant flow, but one that we kind of came up with where you've got a channel model in NeurML2, you have some scripts that actually run that model and come out with some data that's simulated. We compare that with ion channel data from the literature. There's a test, like a SciUnit test, that basically compares these two things. If there's a good fit, we store that in a database. If there's not a good fit, we go back and we optimize the parameters and we try it again. So it's a bit of a simplified flow, but just to give you a sense of what folks have been doing on the channel worm side, and you can substitute this out for a muscle model, you can substitute this out for a cell model, or you can substitute this out for a network as well. This general flow is kind of what we're trying to do right now in the project. Okay, and also excitingly, although it's kind of been a little bit more hush-hush because it's still kind of in alpha, but so Vahid and Milad have done awesome work to put together a little a web app that can start to create a workflow for doing this digitizing and creating some models. This is available on the channel worm repo. and it's also up online, so you can, I think the link in the lower side there will let you actually play with it. I'll let them, in a breakout, maybe talk more about where it's at and where it's going and that kind of thing. But suffice to say, it's a way that you can start to encode some of the basic parameters that you need to define the patch clamp figure here. They've actually built in an open source plot digitizer into the interface, so you can do your digitizing inside this interface, and then it gets stored in a database. The different channel models, the database that they have there, can be listed, as well as so the channel type, the experiment, different actions that you can take to work on it. So it's actually rather nice. And that's kind of fitting in here again to this sort of channel worm piece. And so, so both the pipeline to actually make this stuff happen is being built. At the same time, Vahid and Milad felt strongly that they wanted to have a good way to visualize this so they can get other people involved in doing this and not have everybody just have to do it at the command line, which I'm fine with. So, all right. Okay. So I brought this picture back a few times so that hopefully now you guys are kind of getting comfortable with some of the different things that are happening inside this. I just want to remind everybody that we do use GitHub as our rallying point for things. I think pretty much everybody's comfortable with filing issues for stuff, but it does get a little bit more complicated when we've got multiple repos. So one thing just to keep in mind is, if there are questions or if there are problems or if you find things that do not work, feel free to talk about them on the Gitter chat, but also the best thing is to end up filing an actual issue for that, which you can either do on the GitHub interface or you can do in the Waffle board. I do link down here. We do have documentation on docs.openworm, which speaks specifically about best practices for GitHub. So even if you're a GitHub power user, you could still probably learn something here. So I would say check that out. I also just want to do a brief tour of the repos. So PyOpenWorm is at GitHub/openworm/PyOpenWorm. The latest version is available right now. You can do a pip install PyOpenWorm to check it out. So if you want to play with any of that, feel free to do that. There's some examples on the page. There's documentation. Should be ready to go. It also has its own Gitter chat. Then there's C302. So this is the link to C302. It's also linked down here. This is a snapshot of sort of this vision of having multiple levels of description of the network, which is great that we have that. And so I kind of get excited about moving to D, and I think Porrig is correctly sort of more cautious about that. But this level of D, where we have multiple compartment representations of neurons, and we have ion channels sort of populating them, is I think ultimately one of the more exciting parts that we could eventually go to. So that's here at this location. So that's C302. The muscle model is up here at openworm/muscle_model. There's a direct link there. You can get to that and check that piece out as well. Channelworm is up at vahidgh/channelworm, and there's also some docs for that. The docs are actually quite nice, so you can get even a deeper sense of what's going on there with that piece. Okay, now this roadmap, so now kind of getting down to the last bit of this, which is like, okay, Steven, so this is great vision, but I still need to know actually what to actually work on. So we've been prototyping this Trello board here. We're thinking we may actually move it also into Waffle, but for the moment it's up on Trello, so this is very, very new. So the idea of this is to hopefully have at one glance kind of what's going on across the entire project. We've tried to do this in GitHub in the past, and our milestones have kind of gotten a little stale in that particular repo. So we're trying this again. This is a public board. You should be able to check it out. And basically, for our purposes, this one list, muscle neuron channel, is a summary of all the milestones. And a milestone is a much higher thing than a GitHub issue. So mostly, we should be creating issues, not milestones, because milestones are kind of, like I said, like a whole project to do. But for now, you can go to this board, and you can scroll through this list, okay? And what you'll see in this list is, so at the top of the board, there's some quick information for contacts, for folks that are coordinating the projects. There is a link to docs. There's a link to the organized Waffle board. there's various chats as well that are associated with this. And then each card is going to be talking about a specific repo, and it's going to have a one-to-one relationship. This card is going to have a one-to-one relationship with a milestone that's actually in GitHub. So the key thing here for you to do is to click on a card that has a description that's interesting to you, and then click on both of these links. Okay. So if you click on the GitHub link, you're going to go to GitHub and you're going to see the milestone described in words, hopefully with a checklist, with a sense of like what it means to do that thing. And we've been working hard, and Travis has been working hard to make sure that these milestones actually define like when they're actually done, so what it means for them to be complete. And the other link here is a link to Waffle. And if you click to this, you're not just going to get the Waffle board, but you're going to get the Waffle board filtered by specifically this milestone, which is actually really nice. It'll show you just those issues that are related to completing this one milestone. And then that's a way where you can start to engage on that stuff. If you'd like, you can sign up to Trello and you can comment on the milestone itself, because there's really no other way to comment on the whole milestone. Unfortunately, GitHub does not let you comment on milestones. But just do keep a mind towards, if your question is really just about one issue within the milestone, keep that on GitHub and put it on a specific issue that's there. Okay, and then this Waffle board really is, I think, the powerhouse of this. So as Travis was saying, so this is the board right now for this project. It's under Vahid GH ChannelWorm, but it's not only ChannelWorm issues. So issues that appear on this list are actually going to be from PyOpenWorm and from C302 and even from the Hodgkin-Huxley tutorial all show up here. So so you don't have to go to multiple boards. In one board, you get all the repos. But you can filter the board. So that same way that you can click on a Trello card and filter down and get to an issue, you can do that here in Waffle by using this little filter button and then clicking on this milestones thing, and then you can actually pull up an individual milestone, and then it'll just give you, like I said, just those cards that are related to that particular issue. So you can start to see, okay, so Travis is signed up to do this one, but all of these don't have anybody signed up to it yet. So if you log in with GitHub, you'll be able to do things like comment and sign up and move and make some progress with that. So that's definitely going to be a key resource for us today. Okay, the last slide I think is just kind of a summary and wrap up. So let me break there. Thanks guys for listening to me. If you're not all asleep, so hopefully. The idea is that you've gotten a big picture view of where we're going from the 90,000 foot view of what OpenWorm is about. We've gotten some specifics about why cell modeling, what we're doing here, is important. Hopefully you've gotten caught up a bit on some of the things that are going on with channel modeling. We focused on that roadmap quite a bit so you can see some of the different components. So the task now is going to be to do some breakouts, to get more specific on some pieces of the project that you're excited about, and get to some hacking. So with that, let me turn it over to Travis to tell me what's been going on since I've been doing this presentation.
Okay, yeah. All right, awesome. So yeah, as Stephen just said, the next part is going to be getting to the actual hacking. So I see that a lot of people have signed up for different things under the Birds of a Feather interest list. And if you haven't had a chance yet, the link is already in the OpenWorm Gitter chat, yeah, so you should be able to go there. It's at the bottom of the page, and you can sign up. So, just taking a look here, it looks like, all right, so there looks to be about 7 or 8 different projects that people want to work on. That might expand a little, but basically, we're going to break out into different Gitter chats right now. So I guess I can just tell you guys which Gitter chat to go to for each of those projects. So I can start at the top here, and also keep a window open for the OpenWorm/OpenWorm, the general chat as well. That's where we'll be like announcing sort of what's going on and when to come back, and when we're going to regroup. Okay.
Yeah, and I would say as well, as we're shifting into this, so the next steps of this are going to be at the very end of this, in the last 45 minutes of our time slot here. So I guess that's not 1 hour from now, like an hour and half from now or so, we'll get back and sort of share what we've been up to. In the meantime, if you're in the Hangout now, you can feel free to stay in the Hangout, and you can like mute yourself or video mute yourself if you like. But it's also just nice to like know that folks are kind of here at the same time. But we'll be doing as much as we can, I think, on Gitter Chat, right? Right, Travis?
That's right.
Okay.
Yeah. Okay, so I'm actually going to type out the chat that you should go to, and I'll link it as well on the Birds of a Feather list. So if you go to that list now, so for example, the topmost one there is extraction of ion channel data from literature. That chat should be taking place in the channel worm chat. That's the repo that's definitely most relevant to that. So I've just typed out channel worm chat there for now, and I'm going to make that a hyperlink, and I'll do this for each of those tasks. So
I'll link it. You
go ahead. Link it?
Come out, and I'll do the linking while you're doing that.
Sure. Thanks, Steven. Okay. So we got C302. So that's definitely going to be the C302 chat. Okay.
Is there a C302 chat?
Probably not. Is there a C. elegans NeuroML one?
No. I'm happy to wait here and discuss that with anybody on the Hangout if people want to discuss that here.
Okay. So I'll keep that. I'll write that. Yeah. So C302 chat. I'll change that to. Okay. Solunit test and integration. So that cuts across numerous repos. Assign that to channel worm. Okay, muscle modeling. Okay, awesome. So that seems to be all the Birds of a Feather groups. So I'm not on all these Gitter chats right now, but I guess so as you guys go there, just introduce yourselves, and Stephen and I will come around and see how everything's progressing. So let's see.
Yeah, one thing I would suggest in terms of priority, so it looks like the most number of people are signed up under SciUnit tests and integration.
Mm-hmm.
So maybe, Travis, you want to take that as the starting point, and we just want to take that as the first thing.
Sure.
Because it's just the most populated. And then some of the ones that are a bit further down, I noticed are kind of only single people. We might leave those a little bit till later or incorporate them. So I'm just going to reorder these things here a little bit. And then let me just see. Okay, so that's the most. And there's two. Okay, and muscle model ion channel modeling also has 3. Okay, so I will go into the muscle model one.
Okay.
And I'll be over there. And then all the other ones have either 2 or 1. So, Porig, where are you? I don't see you on the list anywhere.
Can we kill this this one? The C302 one, because that's the same as what's up top.
Is it the same?
Isn't it? It has the same people and the same like this. This one is the same as. Sorry, you can't see what I'm highlighting probably.
No, I can.
Oh, this one is the same as this one, right?
Yes.
So you want the channel worm chat?
Yeah, I think so.
Okay.
Okay, yeah. Yeah, well, and it looks like stuff's going to be happening on channel. It's either happening on channel worm or it's happening on muscle model. It's just a question of like what's the topic.
Right.
So, okay, cool. All right, guys. Well, so hopefully everybody knows in the chat there. I'm going to do a mute here, but we'll come back at that time, and yeah, let's get hacking.
Right. Okay. Yes.
Hi. I just wanted to say, yeah, thanks for adding some of those issues with the in the C. elegans and muscle model and so on. I haven't had a chance to look through some of them, but I will hopefully have a chance next week because I've got a lot of issues myself and a kind of parallel things going on. So I wanted to try to update those. One of the things I mentioned was that you had added some issues for psi unit and nose test based validation of muscle model and C02 and so on.
That's right.
That probably would be good to have a look at. I mean, because there are a lot of tests at the moment around this open source brain model validation framework.
Right.
And so a lot of what's there already is being tested to a certain extent, and for some of those, like running the models, installing JNeuronML, installing Neuron, they need something like OMV, and it's integrated with Travis CI. So I mean,
Yep.
a lot of there is a lot of testing there at the moment. So I think not all of the kind of SciUnit / nose test framework is needed or appropriate for some of it.
So, sorry, my computer just cut out in the last like 5 seconds.
Yeah, I'm just saying that, okay, so yes, a lot of tests are there already. They could be incorporated, but not everything needs new tests or needs to be altered to the other type of testing frameworks.
Right. Okay, yeah. So you're saying we don't need to kind of port over that exact testing framework. We can just kind of integrate what we got there with Travis.
Yeah. Again, I mean, okay, maybe a very light kind of thin wrapper can be put on top to integrate it with some kind of nose test. But again, a lot of these require certain simulators installed to run, and that's what the OMV framework actually does.
Okay.
So, yeah. Okay. But I mean, again, next week I'll try to have a look through them and try to get update some of them. And
Okay, perfect.
the other small thing was that for C. elegans NeuroML repository, a few of them were related to this issue of the two spreadsheets. So there were two different spreadsheets which contain connectivity data, and until we know which one of those is more correct and which one has been used in OpenWorm, I think that's the more kind of higher level issue to resolve before saying, okay, well, let's have a look what's in C302, let's have a look what's in OpenWorm. It should be better to say that I think those are the kind of the raw data, and we just need to be absolutely sure that those spreadsheets contain the raw data, and then that data from the spreadsheet is in OpenWorm, and then it will eventually be in C302 as well. So it's starting figuring out which spreadsheets are correct, where they're from, and having a summary of that in the online documentation.
Right. Okay. Yeah. So Steve and I, as you've seen, like went crazy with raising issues, kinda. So there's like a lot of different issues, but I mean, those are some are short term, some are medium term, and some kind of depend on others. But they're more or less mostly placeholders for now. But yeah, I mean, whichever ones you get a chance to, or like these ones that that we're talking about now, I guess like just throwing a comment on there so we can have some archive of our conversation, because it gets like super complicated. Hey, like.
Yeah, no, that's fine. Yeah. But all of them are generally in the right direction. But it's just some of them are done, some of them are depend on others. But I'll try to comment on them.
Okay, perfect. And in terms of like a C302 chat, do you think we should open one of those? Because I don't, like you said, there is no Gitter.
Yeah,
chat right now.
I mean, I'm much more comfortable working through the issues simply because from day to day I don't know how much time I'll actually have. So the chats are much more kind of fire and forget kind of thing as far as I can see. So I'm happy with dealing with any questions through issues.
Gotcha.
And also, the one problem is that it's still a bit of a work in progress at the moment, and it's a moving target. So, I mean, I'm certainly happy to try to update some of the readmes, but in a week it could be changed, and especially with the optimizer framework.
Totally.
But I'm hoping to try to get more concrete milestones, and then maybe release to people, get people to try. Try modifying things, but yeah, hopefully I'll get some time for it next week.
Okay, perfect.
Hello, Travis.
Hey, it's Triple D.
Hello.
Hello, Sure Joy. Can you hear me?
Hey. Yes.
Okay. All right. Yeah. So I guess everyone else can hear us as well. But if this is annoying you guys, you can just mute the Hangout for a minute. I'm just going to explain the workflow from paper to ion channel model to Srijoy.
Yeah. Thanks.
Okay. I'm just going to share my screen.
Yeah.
Okay. Yeah. So you've seen the
Yeah.
and you are able to access all of this stuff, right?
Yeah, yeah. I was just going through some of the papers and going through the annotations that you guys have made, and I just wanted to know like how that like linked up to the Channel Worm web app that I was just shown.
Totally. Okay. Yeah. So we start just by scanning the literature, and this is something that Vahid has done.
Yeah.
And he's compiled like a number of papers about. I think these are all voltage gated ion channels, and
right.
Yep. So you can go in. So let's just say we've picked a paper now. Let's go to KV1.
Okay.
Okay. So we go in here. Expand this window as well. And we have a paper. We find a plot in here that looks like it would be useful. So something like.
Yeah, that's good. Yeah.
Right. So like an IV curve like this.
Yeah.
And a lot of these as well are based on mutant studies. So
yeah.
We've kind of been ignoring a lot of the mutant data and just looking at the wild type.
No, that makes sense. Yeah, like I do the same thing with neuroelectro.
Oh, okay. So you're familiar with this, like, and then the digitization process, right?
Yeah. No, I guess I'm familiar with like that idea. You kind of just want the control, and then,
Totally.
Yeah. But I'm really interested in like how you. Well, I mean, I guess it's pretty obvious from a plot like this. Like, this is the kind that you'd want. Just show me like how would you then turn this into something that you digitize that goes into. Show me like.
Okay. Yeah. I mean, it's very like primitive the way that we like get it. We just kind of like cut it, right? Like.
Okay. Yeah.
Just cut it straight from the paper. Just get as high res a copy as you can. And a lot of times these papers have figures online, as you know. So those are usually a lot better. And then when we have that image, then the idea is that we can go to web app. So it might take a second to load up there.
Sure.
Okay. And once we're here, we can. Okay.
Oh, okay. Yeah, you're. Hold on, because it was working on my machine. Maybe you have well, I don't know. It's okay. I mean, like I understand that, like.
Yeah. Okay. I've got some pictures of this in the slide, so that's good.
Okay.
So that kind of saves me here.
Yeah.
All right. So we've got. So these are basically the four things you can do right now.
Yeah.
So you'd go into graph mode.
Yeah.
When you click graph, when that gets fixed, it'll bring you to this page where you have the option to upload the image.
Okay.
So you upload it, and then you can. I think there's an automatic option, but the way I've been doing it is manually like clicking on each point in the graph. So I'll go back to this. I click on, let's say this black circle is the control. I click on each point. It maps a point there. Then you can arrange some axes, kind of overlaid, and then give it a scale, and it digitizes for you and creates some points in like a CSV style file.
Okay.
Right?
Yeah.
So that's kind of how we get the plots.
Yeah.
Into like a machine readable format. As for going from there to a model, we'd create a patch clamp experiment, which is just like a data structure that we made.
I see. So, yeah. Okay. Cool. So like, okay. So basically, like the patch clamp data model is to like capture or like represent the data from the thing that you just digitized, right?
Absolutely, exactly.
Yeah. Okay.
And then
And then
that'll
you
be attached to
fill
the
in,
actual story.
yeah, you fill in all those things like either drop down fields or like whatever. It looks like it's just text.
Yep.
And then it's does it like I guess like from all that information then it like recreates this conditions of the not conditions but like I guess then like between the plot and this little bit of like experimental conditions or whatever like protocols like then you have like there's sufficient information to populate a channel model.
Exactly. Yeah. And some of the tools we use, like PyNeuronML, they take a lot of these parameters, along with temperature and some other basic parameters that you mentioned in the chat. So once we have all those parameters, and we can run it on the command line and generate an IV curve or whatever kind of like I guess like just a current over time plot as well, and then we can test those against the one that we digitized from the literature a little while ago.
Yeah. Okay.
Right.
Great.
Does that kind of clear it up?
Yeah. No. It really does. And so let me see. Like, like while I have you on the chat, like I think there's a couple things like I can probably contribute to like more directly. Like.
Okay.
And so like for example, like can we just go to that paper that you were just showing to me?
Yes.
Yeah. Okay. So like, are you interested in getting out like if you see like not the plot, but like down here they show like the V50. I can't really read that on your but you know all those numbers down there. Is that something that you're also interested in, or is the data sufficient? Is the plot sufficient for you?
You're talking about this caption here?
Yeah, the caption. Yeah, basically.
Yeah. Okay. Yeah, I see. And it's mentioned over here as well. I think some similar things. Yeah.
That's right.
All of this is interesting to us. Basically, the way that we're going through these papers is sort of at a high level, and we'll basically. So we're referencing all the. If I'm just going to jump to the sheet for a sec.
Okay.
So this is KV1. This was, I believe, the Fawcett paper.
Yeah.
So we'll say like 2C.
Okay.
And we'll come back and see 2C. We haven't taken note of these parameters on the sheet itself, but it's kind of assumed implicitly, which is not good enough. I guess we need to kind of make it more explicit as we go along. But this is basically a first pass. We're going to mark down where we should be looking in these papers when we come back to them, and then when we come back, we'll extract all of the data that comes along with the plot. So most of the time is contained within the caption.
Okay. Yeah. I think that like that all makes sense to me, and like I think if you were, I mean, like I'm just looking down here for many of the things in the caption, like it's just a text representation of what was in the figure. So if you can, for a lot of things, like you could probably just get if you had the figure, then you wouldn't need that stuff that you just highlighted.
Right.
I guess. And then there's other things about like the exact conditions, like the 10 millivolt steps or whatever that goes into your
into the data structure.
Yeah, that's right, into the data structure to help represent the data.
Right.
Okay. I think that's clear.
Right. So I guess in terms of like actions, like if what you're doing right now, we don't really need to list all these parameters because this is just like a first pass. As long as we know that they're there and attached to each of these figures.
Yeah.
So if you notice something, yeah.
Sure. Would you say right now the goal is just identifying or linking papers in graphs with relevancy, and then you'll work on digitizing later?
That's exactly right.
Okay.
Yep. There's actually, okay, yeah, the issue you're working on was number 8, but there's another one that I'll link you to in the chat after this, which is more directly what we're talking about. It's more like collecting these things, and then there's another issue for the actual digitization. So I guess collecting is definitely the step that we're working on right now.
Yeah. Okay. And then like just for like my own research interest is like I'm like obviously like this will work in a really manual way, and like if you guys want, if you're happy with the manual solution, like basically it's like if there's just not that data that you need to extract, then like a manual solution will work. But like if there's like a lot of things and you don't really know how comprehensive your finding approach is, then you may want something a bit more automated or something that's like allowing you to cast a wider net.
Right, totally. So like some kind of data mining thing, is that what you're thinking?
So like a lot of what I did with NeuroElectro was like text mining, and so we'd have like there, like my focus wasn't really ion channels, but it was neuron types, and then trying to
Right.
cast a really wide net to capture things about like electrophysiology properties of different neuron types, like from as many papers as possible.
Right, totally. Yeah, and no, absolutely. And there may be an issue related to that in the channel worm repo, but that is something that we've talked about before.
Yeah.
I guess in terms of like where we are right now, this
Yeah.
is the manual solution is kind of what we have to do first, and basically get from paper to model, do it
Yeah.
a few times, and then we can automate the process. But you're absolutely right. I mean,
Okay.
making it automatic is definitely a step in the future.
Okay, cool. That makes sense. And so yeah, thanks for walking me through this. I think I know what I can do for a little bit.
Okay, no trouble. Yep.
Okay.
All
Okay,
right.
great.
Talk
Thanks,
to you in
Trevor.
chat. Yep.
Okay, bye.
Hey, Shujoy.
Hey.
Thanks for joining us.
You're welcome.
So I guess you got a little bit better sense now for
Yeah.
challenges. It's a process, but yeah, that Channelpedia stuff too is going to be awesome when it's back in.
Yeah.
Did anybody else notice our agenda getting hacked earlier?
Yeah, I was very surprised.
World editable maybe wasn't such a smart move, so we put it back to.
I guess it was bound to happen.
Yeah. So,
oh well, yeah. Well, next time we'll make it. Yeah.
All right.
Welcome back, Rick.
Hi. So what are we waiting for a couple more people?
Yeah, we'll wait until this thing is, and I think it is right now, full. Yeah, I was very surprised.
World editable maybe wasn't such a smart move, so we put it back to. Somebody's on YouTube. Somebody needs to turn off their YouTube.
Oh, well, yeah. Well, next time we'll make it. Yeah.
All right.
Welcome back, Rick.
Hi.
I think it was Gopal. I did that once, and it just completely ruined the Hangout. I couldn't find the tab where the same Hangout that I was on was playing. I couldn't find it, and it was for a general meeting. It was horrible. I had to shut the whole thing down, or I think it was like a journal club. It was just terrible. So, yeah.
Right. So many tabs. Yeah. Okay. Yeah. I think so. I think we're full again now.
Yeah.
Okay. Perfect. So yeah, it looks like we had a lot of activity in like all the chats that I was looking at anyway. So that's awesome. I guess we can just quickly go through everyone in this chat at least and give sort of a short update on what this hackathon kind of spurred into motion. Maybe a lot didn't get done like in the past couple hours, and maybe it did. But in any case, just tell us, I guess, what's changed and kind of what the next steps are going to look like for your particular problem that you decided to tackle here today. Okay, so we'll start off with Vahid. He's the closest to my image on my screen. So Vahid, if you want to give a quick update, I don't know if there's still bandwidth issues, but we can give it a shot and see. Vahid?
I think he's muted.
Yep. Yeah, Vahid. Okay, we'll skip Vahid for right now and go straight to Srijoy.
Okay, so I was going through I was working on trying to get data out of papers on channels into channel, I guess like the ChannelWorm web app, and so I was just going through like a lot of the Google Docs like tables and looking to see like how the information was encoded there to start, and then I think I noticed that like what you guys have, like it seems like a really good start as a way of encoding the information, and then but A lot of what me and Rick learned through Neurolecture, I think, could help, like maybe go from like these really disorganized, or maybe pseudo-organized spreadsheets to something a bit more organized, and thinking about like a data flow pipeline. And so I think one thing that would go a long way is just coming up with some pretty simple guidelines for things to look for in these papers, and then making it clear to a curator like what they're looking for, how it should be encoded. And so then it would just help, like, it would just help the onboarding process for people like me who want to work a little bit on getting channel data out of papers. And so I think me and Vahid and Travis we'll have a quick meeting on that later this week.
And you'll drive yourself crazy with like schema migrations and stuff early on as you try to figure out what it is that belongs in your database and what fields and everything. But then when you get through that, it's actually much more organized at the end.
I think at this stage it's good to start with spreadsheets, and then maybe when that begins to crystallize, you can start to think about how you encode that into a MySQL database or into Django. But maybe doing that really early on is maybe not so helpful. Otherwise, you'll have the schema migration problem.
Totally. No, that makes a lot of sense, actually. Okay, yeah. So, there'll definitely be like a lot of discussion about that too in this meeting that we have in a few days. Okay, perfect. Yeah, thanks, Srijoy. Rick, do you have anything to add to that, and also what you've been doing?
Let me just screen share some code I was writing during this time.
Okay.
After I took a brief break and was working on a puppet show with my daughter and my wife and my son. So let me then I got back to coding. So let's see. Let me get to the screen sharing app here. I think it's this one. All right, let me see if I can. There we go. All right. So this is that notebook that I had was in the ChannelWorm tests directory, and first there was just some. Those of you who haven't seen it, this is just some like boilerplate SciUnit stuff that I had in there just to kind of show how it works. But I want to replace all of this with stuff that's relevant to the channel model, and just move this kind of stuff that you see here back to sort of some documentation. But basically, what I've been doing for running models and then testing the results was, just importing the basic modules, including this is from Porg's PyNeuromL project. There's this NML2 channel analysis, which is basically has been a script, but I've been sort of reworking it into a bunch of more focused functions that we can use, and then there's okay, so you want to run the channel, so you have your channel file that describes the mechanics of the channel, and then you actually have your parameters for running it, which are like the temperature and how what you want your voltage steps to be and so on. And not everything in the script is actually useful. For example, if you're just doing iterative model fitting, you don't need the graphics and you don't need some other stuff. A bunch of so many files to be written. So basically, what I was using then was this ChannelWorm CW fitter thing that I think Vahid was working on, and that one of the things it does is it takes something from a figure in a journal article and just extracts some parameters, and then so you can sort of just compute IV curves and basically plot them. So to get them on the same scale, I was just interpolating so they had the same range of voltages, and then the resulting plot was here, which looks terrible. The black is the data, and the red is the model prediction, and that looks pretty poor. I think there's just a scale issue here, among other things. But that's an issue right now to be solved. But and then there's sort of a print of an error metric, which is in this case is a mean squared error of current in the IV curve. But what I worked on today was I wanted to get all those things out of the NML2 channel analysis and get them into SciUnit form. So there's some basic defaults for temperature and all that stuff, and so in SciUnit you have to make these capabilities. So I want to say, okay, one of the things we can do is we can run the models, and the other thing we do is we can produce IV curves and plot them, and then the model itself, the SciUnit model class, which is going to take care of running and executing these capabilities against the model, is here, just having, okay, there's this model. It takes a NeuronML channel file, and which may have one or more channels in it, but we want to try to keep each model to one channel, and then it's just going to, just extract a few things. These are all new functions in NML channel analysis that are that I made that used to be just little loops inside this script, but I've sort of taken them out into functions now, so I can use them. And then basically there's this sort of run, which is to say make the lens file and run it, and then produce IV curves and plot the curves. So then essentially what then it looks like from the point of view of someone running SciUnit then is, okay, there's a model name, there's a file path, and there's instantiate your channel model, okay, and it has a name. And then the next thing to do, which I didn't get to, would be actually running these functions. For example, just produce IV curve steady state, okay, which is going to be a steady state IV curve thing. It's going to just produce some data, and it's going to set it to an attribute in the class, and then eventually you would call this plot. Now, you don't need to call the plot. That's sort of a convenience function. The actual in SciUnit, you're going to have rather than actually do the plotting, it's just going to do sort of this kind of interpolation up here, and then compute a goodness of fit metric and return that to you. But that's kind of where I got. It just was essentially taking stuff that was in NML2 channel analysis and breaking it down and putting it into a form that's easier to work with from within Python scripts and functions running in loops and all that stuff. So
awesome. Yeah, no, that looks like a lot of good progress today. Okay, so just to be clear, is that stuff down below the stuff that you worked on today?
Yeah.
Is that calling the previous things that you wrote up above the graph?
Not yet. So what it's doing is this is all new. This is all me using new pieces of NML2 channel analysis that were in Porg's script. I mean, I didn't really change, except for a little bit of cleanup. I essentially changed very little in what was in that script. I just broke it all down into pieces that I could use, and then otherwise it's right now just getting to the point where it runs the model. And the next thing in here is that is to write the test in SciUnit as well. So this is what the model looks like in SciUnit, and then the test, there's going to be a whatever IV test or something, and it's going to have something that's going to say, to compute this and that, and it's going to, or get prediction, for example. Get prediction is going to be a function that's going to call SciUnit stuff. It's going to call the model. Essentially, it's going to call the model to run, and it's going to then having gotten a prediction, then there's going to be something below it. It's going to be, these are all standard SciUnit things that have these names. It's it's compute, basically this function that computes the agreement, and it's going to take that prediction, and it's going to take the observation from that's generated up here from extracting from the data from CW Fitter, and then it's going to do the comparison that's basically here. For example, it doesn't have to be that comparison. That's just one way to do it. Then it would return a score. So I didn't get to the scoring part of it today. I just got to the to the generation of the model and being able to run the model through SciUnit.
Right. That's awesome. Yeah. Okay. That looks great. And as for that issue with the graph, that is an issue in GitHub right now, right?
Yes, I just put it up.
Excellent. Okay. Perfect. Thanks, Rick.
Okay. Awesome.
Okay, Stephen. Well, Stephen and I were both basically going around sort of curating the chats and making sure that people were able to get the resources that they wanted. So, Stephen, I don't know if you wanted to give an update on what happened there.
Yeah, I was just sort of pointing folks at each other and at resources. I did make a new milestone while we were talking that just wraps up some of the performance issues, because Abdullah ran into some known PyOpenWorm performance issues, and so that's one thing I think that we can start to address. So maybe just a milestone that wraps that up, sort of saying that all functions in PyOpenWorm should return in 1 second or less. So we try to get creative for that. But yeah, other than that, just pointing people at stuff.
Okay, perfect. Thanks. And now, Milad, if you can hear me, if your bandwidth's okay, can you give us a little update on what you've been doing today? may have the connection issue with Milad as well. Okay, so we'll come back to Milad if his connection fixes up. If your connection comes back, just send us a message in the chat there. Okay, Joe. Joe, you're on mute. Can you hear me? Okay, it looks like Joe is unavailable as well. Okay, so we'll jump to Gopal then. What have you been doing today, Gopal?
So I wanted to just finish off the PyOpenWorm test refactoring that you and I had started about a month ago, and I had completely forgotten about one. We may not have made an issue for it at the time, but the whole class of data tests that we ended up not refactoring out because I think when we first tried it, we ran into some dependency issue, and so we figured we'd just get back to it. And so I got started on that, and for whatever reason, all of the tests were failing on my machine, but it looks like I resolved that. So I'll just get back to doing that on my own, and then hopefully they'll just close the book on that sort of test refactoring effort, and then we can move on to other things.
Okay, awesome. Thanks, Gopal.
Cool.
Okay, yeah. So that's everyone in the chat. It seems so. Milad message and Vahid message, and they're both basically having connection issues. So if you guys can give an update on what you've done today in the Gitter chat, I think both of you guys would be in Channel Worm. Just like a couple sentences or whatever, and we can also go back through the logs and see kind of what occurred. But yeah, so that's everybody in the chat right now. Stephen, were there any words you wanted to have before we kind of wrap this up here?
Well, I think we should probably talk about next steps now.
Yes. Okay, yeah. And also next, I guess the next time we'll meet. So Steve and I have been discussing privately. How are we gonna?
Did Abdullah get a chance to update, actually?
Oh, okay. No, I didn't see Abdullah there. Okay.
Abdullah also wrote something in the chat recently, but do you want to say it out loud?
Sure. I was working on a script to pull data out of PyOpenWorm and use it in C302. MWatts has already done that before. So I used his script as a stub and updated it to use the latest version of PyOpenWorm, and I've uploaded it as a gist, and I've referenced it in the chat room. I'm going to push it to my fork and then do a pull request after a certain issue about which data source to use which has been raised by Porig would be resolved.
Awesome. Okay, thanks, Abdullah. That's great. So this was on the C302 repo, right? Yes. Okay. All right. Yeah. So next steps. Stephen, you were saying something.
Yeah. So guys, so the idea of this hackathon was, as much for everybody to kind of get a sense of what's going on, but also to kind of reboot the sessions on this stuff. So the idea is not to just do this and have another one in 3 months, but to actually establish a rhythm here. And so Travis is with us at least till August 24th. So we can definitely keep a pace up on that, and we can see what rhythm we can keep up after that. So I think the proposal is now to not lose momentum. You guys have kind of gotten re-engaged on a few different things, and what's worked really well in other parts of the project are to have some standing meetings to sort of check in on progress at a time that it's sort of predictable, that everybody kind of knows when it is. And I don't think we know what that is for everybody here, what that would be, but I think the next step is to send out a poll to figure out when we could get together. And the form of that get together, so Travis and I were kind of kicking it around. So this whole hackathon thing is sort of new. So I think that one question that we have is like, do we want to do this as a check-in where we kind of go around and talk about what we have done, or do we want to make it a practical session where every 2 weeks we kind of get together and hack a bit? We could update kind of quickly at the beginning, but that the focus of it is a hackathon. So one question might be like, are folks interested in a 2 or 3 hour every 2 week shared hack session kind of like what we did today, except without my lengthy intro, obviously, at the beginning. You've got that. And do that every 2 weeks between now and the 24th of August at least, and maybe see if we even continue it after that too. Would folks be down for something like that?
Well, yeah, of course. Yeah, I am, of course. So, what about? Okay, Gopal looks like he's unmuted.
Yeah, that would be great for me, and I have about 3 weeks that I would like to work on this full time, starting next Monday, the 29th, through July 20th. And so I'd like to get as much done as I possibly can in that 3-week period. So then after that, I'm imagining 5 to 10 hours a week once school starts for me.
Excellent. Okay, Rick, you're off mute as well.
I'll probably be available, once a week or whatever.
Perfect. Srijoy, I think we're getting a consensus here. Oh, you're on mute, Srijoy.
I want to be available as needed.
Perfect.
That's also so that's also totally doable. Like you guys were all using Gitter chat today. It's a very nice way to be able to mention folks and stay in touch. So it's definitely also doable.
Right. Yeah. That's a great way to keep up with everything asynchronous, sort of asynchronously as well, especially with the mentions and stuff. Okay, looks like Porg is back as well now. Hello.
Okay.
Yeah, we were just talking about sort of doing this again every couple of weeks. So I don't know what your schedule's like, Porg, or what, but you just came in at the right time to kind of give your input on further hackathon sessions. What would you think of that?
Sundays are difficult. Many other things to do, but I think maybe once a month, but if possible, maybe smaller, kind of quicker sessions during the week. But
yeah. Okay, awesome. All right, so I guess that kind of wraps it up now. What do you think, Stephen? Are there any other next steps that we can take care of here?
Yeah. So the final answer to this will happen over email. Early next week. Plus, right, any other ad hoc sessions that you guys want to have, obviously, have them. That'd be great. And I just wanted to express my thanks to everybody who took the time out here to get together and work on this. It's not really for me. It is, I think, in favor of this project, which I think is really important. I think it's something that's very timely. As you can see from my intro, there's a lot of unanswered questions in this space, but it's something that is actually possible for us to address as a team. So it depends on each and every one of you working on it, as well as the combination of your efforts with the other sub-projects and the other folks that are working on the other aspects of the project too. My hope is that before too long, we're actually able to get that entire group together for a mega, ultra, super duper hackathon. So then we have cross-pollination with movement validation folks and cross-pollination with all the Geppetto folks. I kind of wanted to have some of these sessions as a way to build up to that. But maybe at the end of the summer or something, we could do like a super meeting, and obviously we have to solve problems like how many people fit in a single hangout kind of thing. But those are solvable. So anyway, yeah, just really great to see everybody come out, especially folks who I know have been sort of very patiently waiting to get updated with new stuff. I hope you feel like you have now not just one thing to do, but you have a bit of a roadmap for seeing like all the milestones and seeing kind of the path in general as we go forward, so that you can pick up something maybe that I have not directly told you about, or Trevor's not directly told you about, but that you kind of see maybe where it fits in the big picture. That's really what's needed for us to make this thing self-sustaining, is for all of us to be able to do that process. So I'll wrap there. Thank you very much, and this was really great. This was a really great session.
Thanks so much, guys.
Yeah, thank you, guys.
Okay, see you later.
All right, everybody.
Nice meeting you guys. Bye.
Bye-bye.
Bye-bye.